Okay, good morning, everyone. Thank you all for coming. I would also like to thank the organization for giving me this opportunity to present our work. Lord, sorry, I'm shorter than all the other speakers. So my name is Chelsea Zhu, and I'm a PhD candidate from UCLA, graduating in September. So for today's presentation, I'm going to introduce a deep learning method to predict different properties of the protein-protein interaction. So the problem that we're interested in can be formulated as, given a pair of protein sequence, we wanted to know whether these two proteins interact or not. If they interact, what is the type of interaction? And what is the strength of the interaction? So to give a little bit of background on why those problems are very important, the knowledge and information extracted from the protein-protein interactions are essentials toward understanding the cellular and biological processes. And they can also empower our understanding on biochemical cascade of different disease. And understanding the protein-protein interaction can also facilitate the putative therapeutic targets identification, which further facilitate drug development and drug design. Existing high-throughput computational methods can be divided into two categories. The first one is the traditional approach that uses statistical learning. And the second one is recent development of deep learning frameworks. In statistical learning, most of the effort focuses on feature engineering and feature selection. These features often either summarizing or aggregating the physical-chemical properties of the amino acid. And based on the selected features, one of the learning algorithm is applied for prediction. One of the problems is that the process for feature engineering or feature selection is kind of costly. And more importantly, most of the selected features only cover a very specific aspect of the protein profile, and they often provide a very limited coverage on the interaction information. And to address this problem, several deep learning algorithms have been proposed to automatically learn the latent features from a protein sequence. DPPI and DNN-PPI are two of the methods along this line, and both of these methods use a deep convolution neural network to automatically extract the significant local features from a sequence. However, the CNN-based framework often only focus on the local feature, and they cannot capture the contextual and sequential information from a sequence. In addition, DPPI uses CyBLAST to construct the protein profile, which is actually an extra step for data preprocessing. So we present PIPR. This is a deep learning-based method that incorporates a residual recurrent convolution neural network. The idea is that we want to build a system that is able to learn those important features from the scratch. And this elevates the feature engineering process and also removes the needs for data preprocessing. Secondly, we also want the system to be able to capture the contextual and sequential feature of a protein sequence. Lastly, we want a system to be able to predict different types of information. For example, like the classification task or the regression task. So here is actually a diagram of the overall schema for our framework. It started with a property-aware lexicon encoding system to represent the amino acid. And the embedding of the amino acid is transformed into a sequence embedding through a residual RCNN network. Finally, we apply a Siamese architecture to learn different types of tasks. And in the next few slides, I'm going to talk about each component in detail. To represent an amino acid, we use a semi-latent encoding schema to capture both of the contextual and physical relatedness of the amino acids. So the first 5 latent space describe the contextual properties of the amino acid. The idea is that if two amino acids co-occur frequently, they should be closer in their embedding space. And we use a script-web model to measure the co-occurrences of the amino acid. And this is pre-trained on 8,000 protein sequence obtained from a database. The second part of the embedding system is describe the physical-chemical property of the amino acid. Each amino acid can be clustered into one of the 7 classes based on the diploid or volume of the side chain. And we use a one-hot vector here to capture this information. Next, we want to represent the protein. And this is done by converting the amino acid embedding into a sequence embedding. We use a recurrent convolution neural network to do that. And the goal here is to aggregate the multi-granular features from a raw sequence. So the pre-trained amino acid embedding is fed into a recurrent neural network. And since this is a recurrent structure, it contains several layers. And the results from the final layers is fed into another convolution layer, followed by global average pooling to generate the protein sequence embedding. And inside this RCNN unit, it contains several components, including the convolution layer and the maximum pooling mechanism, also the bidirectional gated recurrent unit with the residual shortcuts. So the convolution layer aims at capturing the local feature of a sequence. The input to the convolution layer is either the pre-trained amino acid embedding or the output from the previous neural layer. And the mechanism is done by applying a sliding window of size h on a protein sequence to aggregate the information to combine the features of the neighboring amino acid. And the output of this convolution layer is a sequence of latent factors summarizing all the local features of this H-gram. And the combined features is further summarizing using a maximum pooling mechanism. This is done by taking the maximum value along each dimension of every non-overlapping end stride. This serves for 2 purposes. One is to discretize the convolution results. The other one is to preserve the most significant feature within each end stride. So to preserve the local information over a very long sequence, we use a bidirectional gated recurrent unit to retain the critical information. The GRU contains 2 types of gates. One is the reset gate, the other one is the update gates. And these 2 gates control the amount of information flow from the past to the future. The input to the GRU is the polling results from the previous convolution layer. And we apply a bidirectional GRU to capture the sequential information from both directions. The last component of the RCNN unit is the residual shortcuts. This adds the input of the GRU to its output, and it provides 2 advantages. One is to simplify the learning to capture only the differences between the input and the output. The second advantage is to prevent the model from vanishing gradient problem. And to represent this protein pair, we need to convert the sequence embedding into the protein pair embedding. We use the Siamese learning architecture to apprehend the mutual influences of the 2 sequences. And this is done by using the same RCNN encoder for the 2 sequences. The embeddings of the 2 protein sequences are combined using an element-wise multiplication to generate this protein pair embedding, and this protein pair embedding is further fed through a multi-layer perceptron with a leaky ReLU as activation function. This is to model the nonlinear relationship among the latent factors. The output of the MLP is either a vector for classification problem or a scalar for regression task. And we use two different types of learning objectives for different tasks. For classification problem, we use cross entropy as our cost function. In this setting, NLP outputs a probability of each class for each instance, and we use this probability to minimize the loss. In the regression task, we use the mean square loss as our cost function. And in this setting, the MLP output the normalized score for every instance. And the objective function here is to minimize the predicted score with the ground truth. So recall that there are three problems we're interested in. The first one is we want to predict whether two proteins interact or not. And this can be treated as a binary problem. The second question that we're interested in is to predict the type of prediction of the interaction. And this can be modeled as a multi-class classification problem. The third question is to estimate the strength of the interaction. And in this case, we're interested in the binding affinity of the two proteins. And this can be modeled as a regression task. So in the evaluation, we're going to show that PIPR is able to handle all the three types of tasks. And we'll also discuss the advantage of PIPR over other existing methods. So to evaluate the performance of our framework on a binary task, we use a benchmark data set containing equal numbers of positive and negative cases. This is a widely used database by most state-of-the-art algorithms. And for the experimental setting, we conduct a 5-fold cross-validation. We first compare PIPR with other deep learning architectures. DNN PPI and DPPI are two of the existing deep learning algorithms that use deep convolution neural network, which only capture the local features. SRGRU and RCNN are our own implementation that removes different components from PIPR. The performance is evaluated using accuracy, F1 score, and Matthew correlation coefficient. The result shows that PIPR outperforms all the other deep learning architecture, mainly because the Siamese architecture is able to apprehend the mutual influences of a protein pair. And the RCNN unit is able to preserve the contextual and sequential information and leverage this information for prediction. Next, we compare PIPR with other statistical learning algorithms. All of these algorithms use different ways to extract the feature from a protein sequence, followed by a learning algorithm. For example, SVM-AC here use SVM as a learning algorithm and autocovariance for features. Autocovariance accounts for the interaction between amino acids in close proximity. It also considers 7 different physical-chemical properties of the amino acid. Another example here is KNN-CTD, which use k-nearest neighbor as a learning algorithm, and 3 different descriptors to describe different properties of the amino acids. The descriptors here are composition, transition, and distribution. And the results here shows that PIPR outperforms all the other statistical learning algorithms because the deep learning architecture is able to automatically integrate the important features for prediction. And those features are actually better in encapsulating different aspects of the protein profile. The second task that we're interested in is the multi-class classification problem. The goal here is to identify the type of interaction for 2 interacting proteins. And we use the data sets from the string database. This contains 7 different types of interaction, and we randomly select close to 148,000 of cases for training and testing. The experimental setting is done through a 10-fold cross-validation. And since there are fewer efforts have been attempted for this task, so we implement 2 baseline algorithms along with 10 different statistical learning algorithms. Random refers to random guessing of the interaction type. Zero rule predicts the majority cases in the sample. For the statistical learning, we use autocovariance and the CTD descriptors as feature, followed with 5 different learning algorithms. So as we can see from the results here that the deep learning architecture outperformed both of the baseline and the traditional statistical learning algorithms. And among the 3 deep learning architecture, PIPR presents the best accuracy with a 3.8-fold change over the zero rules. And this observation is similar to what we observe in a binary task. The last task is a regression task, which estimates the binding affinity between 2 proteins. We use a data set from the SCAMP database, which contains roughly 3,000 binding affinity changes upon mutation. The experimental setting here is also conducted using a 10-fold cross-validation. Similar to the multi-class classification task, less effort has been carried out for this task, so we implement our own statistical learning algorithm using autocovariance or the CTD descriptors for feature, followed with Bayesian-rich regressor, SVM, random forest, or AdaBoost as the learning algorithms. The performance is evaluated through mean square of error, mean absolute error, and also correlation with the ground truth. And the results here shows that PIPR presents the smallest amount of error with the highest correlation with the ground truth. And we take a closer look at one of the examples that shows a change upon mutation. The blue complex here is a subtilisine precursor, and the red complex here is chymotrypsin inhibitor. We observe a mutation on the chymotrypsin inhibitor, which caused a significant conformational changes as highlighted in the square boxes here in the slides. And this change actually debilitates the affinity, the binding affinity of the 2 proteins. And we show that here, PIPR can actually reflect this change based on 1 single mutation. So in conclusion, we present PIPR, which is an end-to-end framework from a pair of protein sequence to the PPI predictions. This end-to-end framework contains a property-aware amino acid embedding, and we use a residual RCNN unit to generate the sequence embeddings. Also, we use the Siamese architecture to apprehend the mutual influences of a protein pair. PIPR generalizes well in different types of tasks, including the binary prediction, multi-class classification problem, and also the regression problem. The result shows that PIPR outperforms the statistical learning algorithm as it's able to automatically preserve the critical features. It also outperforms the existing learning algorithm, deep learning algorithm because it incorporates the contextual and sequential features. And with that, I would like to thank the people who actually participated in this lab, especially Moha, who is now a postdoc at UPenn. And this is actually a close collaboration among 3 different labs. And thank you so much for your attention.
Hello? Okay. I think to keep things on time, we'll take 1 question. And if our next speaker could go ahead and start setting up.
You mentioned your show pre-trained embeddings. Did you actually try to run the other models with your embeddings and see if they improve?
I have a backup slide for that. That's a good question. So we actually try different types of embedding. We try different parts. So I'll go a little bit. So for example, here, this is the embedding that we're using, which contain both of the contextual properties and then the physical chemical property. And we also take them out, different parts for the embedding, and we compare also with the one hot vector, which is just a 20 dimension for every amino acid. And the result here shows that this actually, like the way we're using it, present the best. It seems like incorporating both of this information helps a little bit, although the difference is actually not too much. I think most of the contribution come from the model, so the embedding is a small part that contributed to the success. Yeah.